Welcome, everyone. Good afternoon to this session, and thanks for your participation. Before we begin, I'd like to announce that we have a mic there in the middle of the room for audience questions, if anybody has any question after the talk. The talk itself, it will be about 20 to 22 minutes, so we'll have the rest of the time for questions. And another way to submit question is through the app. If you have installed the app, you can submit the question. I will be able to see those as well. So I'm your host, Ratnesh Kumar. I'm with the Iowa State University. I'm a graduate of IIT Kanpur from 1987, and I've been at Iowa State for the past 20 years. It is my distinct honor and privilege to introduce our next speaker. He is among the most decorated person I have ever had the opportunity to interact, as we will see ourselves in a minute. Dr. Rakesh Jain is the Andrew Work Cook Professor and Director of the E.L. Steele Laboratories for Tumor Biology at Massachusetts General Hospital in the Harvard Medical School. He is a fellow alum from IIT Kanpur, receiving BTech in chemical engineering in 1972, his master's and PhD degrees in 1974 and 76 respectively from the University of Delaware, also in chemie. He served as professor of chemical engineering at Columbia University from 76 to 78, and later from 78 to 91 at Carnegie Mellon University. Dr. Jain joined his current position at Harvard in 1991. Dr. Jain has received more than 90 awards from engineering and medical societies institutions, more than 90, including a Distinguished Alumnus Award from IIT Kanpur in 94, and later an honorary doctorate in 2015. He has the rarest of rare distinction of being elected to all 4 US National Academies: National Academy of Medicine in 2003, National Academy of Engineering in 2004, National Academy of Sciences in 2009, and then National Academy of Inventors in 2017. He was also elected as a fellow of the American Association of Arts and Sciences in 2008, and in 2013, and that's he's most proud of. We would be proud of any of these, but he is most proud of this. He was awarded the highest scientific honor in the nation, the US National Medal of Science, by President Obama at a White House ceremony. Dr. Jain has mentored more than 250 graduate and postdoctoral students, and his research findings are summarized in nearly 800 publications, which have been cited more than 200,000 times. That's like 200 times more than my citations. He serves or has served on advisory panels to government, industry, academia, and editorial boards for 22 journals, including Nature Reviews Cancer and Nature Reviews Clinical Oncology. Dr. Jain is regarded a pioneer in the area of cancer biology for uncovering the barriers to the delivery and efficacy of molecular, nano, and immunotherapeutics in tumors, developing new strategies to overcome these barriers, and then translating these strategies from bench to bedside. He is most celebrated for proposing the groundbreaking concept that we'll hear about, that the normalization of blood vessels can improve the treatment of various cancers and as well as non-cancerous diseases that afflict more than 500 million people worldwide. He validated his concept in studies in mice and then in patients, leading to FDA approval of multiple drugs for cancer treatment. Let's welcome Dr. Jain to the stage to listen about his journey from chemical engineer to tumor biology and tumor treatment pioneer. With a warm up.
It's such a pleasure and an honor to be back among IITians. after a long time. As Ratnesh mentioned, I did my chemical engineering undergraduate in 1972. And as I was in my senior year, I was contemplating applying to graduate school. And at that time in chemical engineering, the University of Delaware was a leader, courtesy of DuPont's. And many of the textbooks I had used at IIT in chemical engineering were written by professors at the University of Delaware. In particular, the person I really wanted to work with was, when I arrived here in 1972, was Professor Morton M. Dan. And my chemical engineering master's thesis was on mathematical model of controlling pollution in Delaware River. and apparently, my understanding of that model is still used. So that's been more than 50 plus years. So as I was finishing up my master's degree in early 74, my advisor told me if you just stay on one more year, you can easily get a PhD on this, because I had made significant contributions. But at that time, and maybe even today, the Indians had the reputation that they were very good in mathematics, but maybe not so in doing experiments. So I said, okay, let's see if I can test that concept. And so I decided to shop around and for a PhD project. And lo and behold, a new faculty member had joined in at that time, University of Delaware, Professor James Way, who later on would become chairman of chemical engineering at MIT for 13 years, and then later on would become the dean of engineering and science at Princeton University, and he just retired a couple of years ago. So Professor Way said to me, you know what, I have an interesting project for you. And I said, tell me about it. And he said, would you like to study diffusion of drugs in tumors? I said, what? And I said, there's one problem here. I never took a course in biology in high school, or certainly not at IIT at those days. So he said, don't worry about it. And he, by the way, came from Mobil Oil Corporation. He was director of research there when he had joined, just before he joined University of Delaware as an endowed chair. And he said, here's a textbook on human physiology by Guyton. See if it makes sense to you. So I took that book and just hid into my room, and I read it cover to cover. And a week later, I went back to him, hardly any sleep. I said, I think this makes sense. Yeah, it's quite readable book. Later, only Arthur Guyton, who was the author of that book, was a Harvard Medical School graduate. And so I said, okay, so at least you've read the book, it makes sense. Now how about doing one more thing? I said, would you like to go to Washington, D.C. to a National Cancer Institute with me? I said, why? So he said, there's a very distinguished MD there, who's the chief of laboratory pathophysiology, named Pietro M. Gallino. And Gallino, those of you may not know, when cancers grow in our body, they have many arteries, many veins. They have many inputs, many outputs. So, but what he had developed, he had done many beautiful and seminal studies. But one of the things he's known for is he had developed a tumor which had a single artery and a single vein. So I went to his lab and he showed it to me. And I looked at it, I said, this is great. It's like an engineering, input-output analysis. So one input, one output. So I said, could I use this to study drug distribution in tumors? And he said, why would you do that? Drugs always get into tumor, that's why we give them to patients. I said, but I couldn't find any publication. Of course, that was pre-Google days. Still, I couldn't find any. And so after a lot of going back and forth, he said, okay, it's a useless problem, but why don't you work on it if you really want to? And I said, the advantage of that is, once I do input-output analysis, I can apply mathematical modeling and try to figure out what's going on inside the tumor. And then I can get a degree in PhD in chemical engineering. So my advisor and they agreed, and we made a very good agreement. So I would used to go every Sunday night from Delaware. I'd drive to NIH in Bethesda, only a couple-hour drive, and then do my experiments the whole week. And Friday night, I'll go back, and over the weekend, I'll analyze the data. And essentially, 18 months later, I was all done and got my PhD. But the message of my PhD was, drugs don't get into tumors. They are highly heterogeneously distributed. And most of them sit in the margin, and they bypass a tumor like highways around, beltway around a city. So that really perked my interest. So I said, you know what, Dr. Courtesy of Professor Wei, I had a job offer from Mobile Oil Corporation before I finished. And I said, look, I really don't think I want to save money on making gasoline. So I applied to about 150 chemical engineering and bioengineering departments in the country, and for an assistant professorship. And 149 rejected it. And the reason was very simple. The country was going through a recession at that time. So there were not many openings. And even if there were, they said, well, you don't have a green card, so sayonara. And so the only place which invited me was Columbia University, New York. Really, that was not my choice, but I went there anyway because that was the only invitation I got. I gave my talk at 11 a.m., and at 5 p.m., they made me a job offer. I had no choice. I accepted it. And that's where I wanted to ask the question, why is it that drugs don't get into the tumor? And I wrote half a dozen grants to NIH, NSF, everywhere, and they all rejected, saying, with questions like this, not an important question, to what's a chemical engineer doing in cancer research, and things like that. So after 2.5 years, so I did, to sort this problem out, essentially what I did was I did work initially in mathematical models, because all you need is paper and pencil, and really you don't need much money for that. So I did that, and lo and behold, in my second year there, I was approached by chairman of chemical engineering at Carnegie Mellon, saying, would you like to move there? Would you like to move to Carnegie Mellon? I said, look, you had rejected me when I applied in 74 or 75. But nevertheless, I moved there, and that opened up the whole thing. And essentially, what I discovered, and I'll show you very shortly, is the reason drugs don't get into the tumor is because the neighborhood of the tumor, and the medical word for this, microenvironment, is abnormal. It creates barriers. And what I have done in my career is sorted out these very barriers, what they are, how they are imposed, and then what we can do to fix them. And then we've done these studies mathematically in animal models, then taken these ideas to cancer patients and tested those ideas, and that led to some approvals, which I'll show you. So those of you who are not biologists, which I was not in 1974, cancer, when somebody says they have a breast cancer, what does that mean to you? What it means is tumor is not just a collection of cancer cells. Actually, the most deadly tumor is pancreatic cancer, and that has collection, only 5% cells are cancer cells. So what's in 95% in that case? The rest of what 95% is known as microenvironment or stroma. There are many other words for it. For this lecture, I'll call it neighborhood. All right? So what does this neighborhood have? So this neighborhood has is cancer cells, shown here in the gray color. These are the cells which everybody in the field, when I got into, was working on. But these cells, for them to grow, they need to be nourished with blood supply. So the blood vessels are formed. That's called angiogenesis. And so you see the blood vessels in red color. And then excess fluid from the tumor, like normal tissue, needs to be drained, and is done by a set of vessels known as lymphatic vessels. And all these vessels and cells are embedded in extracellular matrix. But in addition to the cancer cell, there are cells of the host, normal cells. And they include immune cells of different flavor. They immune something known as fibroblasts. These are the cells that make matrix. And so what I did is, So just think about it. If you're a drug molecule injected systemically, you're going to go through the vessels, you have to cross the blood vessel wall, go through this matrix, then reach cancer cells, and then eventually leave. And this whole area, this whole neighborhood was not understood at that time. It was a black box. So my mission was to open this black box and try to figure out what we can do to repair things in this neighborhood so that we can deliver drugs and make them more effective. So in the beginning, as I said to you, at Columbia University, I didn't have money, so I did work initially with mathematical models, try to develop models for transport of drugs and tumors. And then, you reach a dead end with mathematical modeling. You need to validate it. You need parameters, values somehow. So what we did is we developed, usually at that time, the tumors were studied by growing a tumor in the mouse or a rat or a rabbit. And then you give your drug and you kill the mouse at a certain time, take the tumor out and see what's going on. But that doesn't tell you what's happening in time, real time. So what we did is we developed glass windows in different places in mice. So this is what is known as dorsal window. It's in the back skin of a mouse. We have windows in the brain, you name it. And what these windows allow us to do, to look at not only drug delivery, but everything and what's going on in real time with microscopy, and you can later on analyze it. And from that you learn things, and then you want to test them in patients. So we did clinical trials, and I've been very fortunate in my career that just in the last 2 decades, we have done about 45 clinical trials, all based on the work which came from our laboratory. But not all drugs work in patients, so then we measure biomarkers and try to get an insight as to why they failed, and then we go back. So this back and forth between bench and bedside, embedded with mathematical modeling, has been the way we have done these studies. So as I said to you, we had to develop special microscopes to do that, a special surgery for creating these windows. And once we implant the window, we bring the animals to microscopic stage. Then by giving appropriate tracers, you can look at different molecules, you can look at different types of cells, you can look at different anatomical geometry like vessels and so on. You can look at function of different things, and you can look at the response of tumor and resistance to drugs. So what do you see through these windows? So this is circa 1992. This is like a picture taken with your smartphone, where you have a 1 centimeter tumor, a 1 centimeter window, and you place on day 0 about 100,000 cancer cells, and they begin to grow. And what you see in this low magnification, just only the large blood vessels. Remember I said to you the tumor attracts vessels so it can grow? And you can see that, and you can see this tumor on day 20 becomes, almost 2/3 the size of the window, right? So we have also developed all sorts of technology to look at it, a higher magnification at different depths and so on and so forth. I'll not go into this in the interest of time. But the big message, the first thing I want you to remember I learned from this work, was that if you take cancer cells from the same petri dish, all right, and implant them in 3 places in a mice, in the breast, these are breast cancer cells, in their brain, because brain metastasis is the last frontier for cancer treatment, and the skin. And then you image these blood vessels. Notice that they look very different, don't they? Same cancer cells going to 3 places are making blood vessels that look very different. And you inject a drug that works beautifully in breast cancer, it doesn't work on brain metastasis. What's going on? So I will go into that question, answer to that during Q&A if you want me to, but the bottom line we learned here is that the local microenvironment shapes the biology of cancer in response to various therapies. Now, but even though these vessels look very different, one thing is very common among all these sites. So, by the way, as we were finishing this work, the criticism was, well, this is all Mickey Mouse work. May not happen in patients. Or it may be happening because you're implanting tumors. What about if the tumors arise spontaneously, like they happen in patients, arise on their own, genetic reasons? So we collaborated with Dr. Raju Kuchalapati, who's a very famous geneticist at Harvard, and he had developed mice that developed tumors in their colon, similar to human tumors. And then we looked at the blood vessels in the colon. They look like a disaster zone too, similar to what I showed you in these windows. But then the criticism came up, okay, well, fine, this is good, but this is not human being. So I had a very good postdoctoral fellow, surgeon, who's now chief of surgery and chair of surgery at University of Chicago, Mitch Posner, came to my lab. And I said, Mitch, let's make vasculature, look at how the human vasculature looks like. So a certain fraction of human colon cancers have a single artery and a single vein. And so I said, Mitch, why don't you inject a monomer into the artery until it comes out of the vein? Then you put a polymer here until it comes out of the vein. And what will happen? This monomer will polymerize. And once it polymerizes, you take this half a kilogram tumor or a pound tumor, put that in KOH solution for a month. It'll eat away everything except for plastic, which was a replica of the blood vessels. And now you can look at it any magnification you want and look at this. What does this remind you of? Doesn't it look similar to this? You know, there are some areas there are no blood vessels, and this other vessels are really screwed up and so on and so forth. I hope the message you get here is vessels are abnormal. But that doesn't stop there. What is the implication of normality? So that becomes clear only when you look at things in real time. And what you find out is that some region of the tumor is very well perfused. That means it's got very good blood flow. Now inject your chemotherapy, it'll get here very nicely. Inject any therapy, it'll get here, it'll kill the cells. But look at this region of the tumor. Shut down. It's like a city with a lot of highways, but they're clogged at the rush hour. But this is not only a rush hour, this is like clogged for a long, long time. So now you cannot deliver drugs. And what is sad is that this is where the most dangerous tumors are, cancer cells are. You take these cancer versus the cancer cell from here, implant them in mice, the one from this region is the most metastatic. These are very angry cells. They love to just take off. Unlike these cells, they're kind of happy cells, they're getting their nutrients. So not only are the blood vessels abnormal, and so are the lymphatic vessels. So when we entered the field, there were no technology available to look at lymphatics in function. So we developed one. So I had a very talented postdoctoral fellow, an MD from Zurich, University of Zurich Medical School, Anders Loy, came to my lab. He said, okay, let's look at lymphatics in the tail of a mouse, and if you inject a dextran molecule, you see these lymphatic vessels light up. So these are not chicken wire mesh. These are actual vessels, the lymphatic vessels in the skin. Now guess what? Now you implant a tumor here in the tail and do the same exercise. What do you see? You don't see anything. Complete darkness inside the tumor. So again, not only are the blood vessels dysfunctional, so are the lymphatics. Now what does that tell you? What that tells you is that if anything is leaking out the blood vessels, in a normal tissue, it'll be drained by the lymphatics. Now here they are not functional. So the fluid pressure, chemical engineering 101, will go up, right? And so when we wrote this, a lot of skepticism, so then we measured it in patients, and then the New York Times did a big article about me, about the pressure barrier in tumors in 1990 or so. Anyway, but those are not only, so now if you have poor blood supply, what happens? You have low oxygen level, low pH, low pressure, and it creates all kinds of problems. If you're not a biologist, these are all very boring, but if you are, essentially what it's doing is creating more abnormal vessels, creating immunosuppression, conferring resistance to every therapy, increasing metastasis, changing metabolism, et cetera, et cetera. Now, think about when students come to my lab, I say to them, look guys, you can work on any of these arrows, figure out what's going on, develop a drug for it, or go back and try to fix the root cause, fix the vessels. And then, suppose each of these drugs costs $100,000 a year, which is a pretty conservative number, you need about $800,000 to fix something which could be fixed by if you could repair the blood vessels. So the question is, how do you do that? So in the interest of time, I'm going to summarize 25 years of work. Shows there are two ways we develop two strategies to repair the blood vessels. One we call normalization of blood vessels using antiangiogenic therapy. Now, the reason this is a very counterintuitive concept is because these drugs were developed to destroy tumor vessels. So when I proposed the idea, let's repair the vessels using a drug which was designed to destroy the blood vessels, I couldn't get my grants funded for 5 years, couldn't get papers published for 5 years. I would go to meetings, and my friends would just turn around, will act like this guy is cuckoo, you know. And so anyway, I'll give you the evidence very soon. But in addition to that, some of the tumor vessels are collapsed, not only by cancer cells, but also by the matrix made by the fibroblasts. And then, so we developed strategies to open this. So how do you do this? So when I was a professor of chemical engineering at Carnegie Mellon, in 1988, I wrote a review article about tumor blood flow. And I asked the question, why are the tumor vessels not functional? So if I'm running over time, just stop me, and I'll just, okay? Just don't worry about it. I don't know, I'm not looking at the times. Okay. We're doing okay? All right, good. So why is tumor blood flow impaired? So suppose you are watering your lawn with a hose, and suddenly water stops flowing. Or what are the possible reasons why? Now, all of you are engineers by training, so you should know the answer to this question. Well, one option is your daughter came and just decided to put her bike on it. And that will stop the flow. And then if you can figure out or convince your daughter, please, could you park your bike somewhere else, then the water will resume. So we have developed equivalent of that in a strategy, and I will not go into the details because I have only a few minutes. But this is the work of about 10 years, essentially. What it shows is the most widely prescribed antihypertensive drug used to control blood pressure, which costs about 14 cents a day, known as losartan, given to 20 million people worldwide. If you give that, you reduce the matrix, and vessels get decompressed, and now you can perfuse drug. And we have shown this in patients. But that's not the only reason. The second reason is that you are watering your lawn, nobody's put their bike on it. What else is the reason why the blood flow or the fluid will stop, flow will stop? Well, one reason is that somebody made a lot of holes in it. So if you make a lot of holes in a tube, the water will stop. Now, what happens in tumors is they make a molecule known as VEGF, vascular endothelial growth factor. And this molecule is required to grow endothelial cells to form new vessels. But one of its key functions of this molecule was originally discovered by my Harvard colleague, Hal Dvorak, vascular permeability factor. So while this molecule is needed to grow vessels, it also makes vessels. And if there's so much of it is sitting in the tumor, it's going to just poke so many holes in there. So the whole pharmaceutical company had spent about 30 years by this time developing drugs that block VEGF. Why? So they could kill the blood vessels, right? It's a growth factor. If you kill it, if there's no blood supply, the tumor will stop. But that didn't happen. Actually, tumors become more angry when you do that. They begin to metastasize more. So I said, maybe what we need to do is use judicious doses of this. Instead of giving large doses of anti-VEGF, just leave a little bit of VEGF there so endothelial cells don't die. And what we essentially, and that'll resume flow. And indeed, this is what we showed. So this is a breast cancer going in a mouse and a breast. And you see this video was taken by an MD PhD student of mine, Harvard MIT student. Every 2 hours, he slept next to the mice in the animal colony. Anyway, he's very successful. So you see these vessels become straighter, narrow, and the flow increases, and now you can deliver drugs and so on and so forth, and drugs respond. But if you give the higher dose, this doesn't happen. What happens is the vessels just die, like the drugs were developed for. And then once we finished the mouse work, the same criticism came up. Is this true in patients? So then we did 40 trials, collaboration with our colleagues at Mass General Hospital and Dana-Farber in rectal cancer, brain cancer, breast cancer, you name it, and different tumors using a series of anti-VEGF drugs. Right? And a message which came out repeatedly was if you could improve the perfusion, meaning improve the blood flow, the patients will live longer. It's just so counterintuitive. And so anyway, these are some of the published work in glioblastoma, which is the most deadly brain tumor, lung cancer, breast cancer. You saw the same thing happening. The blood flow goes up, oxygenation goes up, patients live longer. And now we have seen the same thing with immunotherapy. So as we are finishing this, we asked the question, can we use vascular normalization to improve immunotherapy? And immunotherapy has been one of the biggest breakthroughs in cancer treatment in the last decade. Immunotherapy actually cures patients, unlike chemotherapy that improves survival, that gives you more years to live, but you're never cured. You're never cancer-free. But the immunotherapy you do. But that only works in about under 20% of patients. So what's going on here? How can we improve it? So we've been asking the question. And the answer is that same reason, blood vessels are not functional. So we said, okay, can we combine anti-VEGF therapy, like to normalize vessels, with immune therapy, and does it work? And this is the work published in 2012. And again, in this case, a mouse with a breast cancer, and with this treatment combination, the animal survival doubled. And hundreds of labs have now reproduced this work in different tumor models. But the most gratifying thing for me is that in the FDA in the last 4 or 5 years has approved 7 such combinations for different kinds of cancer: lung cancer, kidney cancer, liver cancer, endometrial cancer. And there are about a couple of hundred trials going on right now, so you can just see with time there are going to be some more approvals. What I want to just leave, the very last example I want to give is that vessels are abnormal not only in cancer, but in about 71 other human diseases. So our idea is in 2001 I put forward was that maybe we can improve the treatment in other diseases. So let me give you an idea of some which has already worked. So a disease called macular degeneration, where the blood vessels become leaky. About 1 million people become blind per year in the US. So anti-VEGF therapy has been approved for that. But there the dose used is lower, and that indeed works, not by killing the vessel, because then you'll go blind, but just repair them. Another disease, there's a benign tumor in the ear called schwannoma. When they develop, you lose your hearing. Sometimes they can be fatal, but hearing loss is the first thing that happens. And when that happens, the only thing they do is surgery and radiation that makes hearing worse. So there was no treatment for it. So I asked my colleague at MGH, I said, hey, why don't we give very low dose of this anti-VEGF drug to these patients? And when we gave it to 10 consecutive patients at Mass General, and 6 of them, the hearing came back in about a week. These people had not heard a word for decades. So you can imagine the joy. And New England Journal of Medicine, which some of you may have heard of, is the #1 journal in the world, they do not publish trials with 10 patients. They publish this. Anyway, because of this, in 2014, this was approved in UK. I will not go on and on. Again, the coronary diseases, plaque rupture occurs because of the same reason. Another one is in tuberculosis. This work is just completed, supported by the Gates Foundation, where again, in tuberculosis, people have been working on to overcome the resistance by the way chemotherapists were working on. What we showed is, are the drugs getting there? And then I don't have time to show you the data. The work is just submitted for publication. The drugs don't get in there. When you combine with this, it does, and hopefully the Gates Foundation will initiate. This is a very impactful work because 1.5 billion people worldwide get TB. Not all of them get active disease, but passive disease. So just want to end my talk by saying that what I hope I will be able to convey to you in as simple terms as I could think of, that abnormal tumor is what the major cause of treatment failure is and metastasis is, and we can repair it by fixing vessels like the way I showed you. These are my colleagues, my postdocs and colleagues, and some collaborators. I showed you earlier. These are my other collaborators. Some of you may know Bob Langer, the Moderna co-founder, and others. He's been my and there are many bunch of Nobel laureates there, so I'll keep going. The Gates Foundation, people who supported this work. And for the last 38 years, I started a course at Carnegie Mellon to teach what I just discussed with you to my chemical engineering graduate students. And that started a course, and now we've been teaching this, a 4-day course since I moved, I continued teaching that after I moved to Harvard. And last year was the 38th annual offering, and this year. So if any of you or your children want to encourage them to get into this area, this is a 4-day course. It's a virtual course now because of COVID. So it used to be in person. And it's been, the faculty members include Nobel laureates, all the leaders in the worldwide. So then these are the people I'm grateful to, foundations, and really the people I'm most grateful to are my 250 graduate students and postdocs with whom I've had the pleasure to work with. So thank you very much, and I'll be happy to answer any questions you have. Thank you.
Hello?
Questions?
Well, thank you very much for such a delightful talk and convergence of engineering with biology in incarnation here. So with that, I guess knowing about engineering cause and effect, it makes this happen. So we should feel proud about that for our training in that domain. So we can open up for questions. I guess we have many questions. The person is standing at you can go ahead.
Hi, Dr. Jain. Thank you very much. My name is Dr. Kansupada. Amazing talk. I just want to understand, as you know, Indians have a lot of cardiovascular disease, and you focus a little bit on your research and application for cardiovascular disease, including heart attack and vascular disease. Can you just give us little knowledge about what are the applications, what are the drugs, and what is the future in your mind? Because everything will be AI and percutaneous in future, and maybe new therapy. Just want to get educated.
It's a very good question you're asking. Actually, I tried to write grants to NIH for that, to use this approach to repair the plaque, which is a major cause. Plaque rupture is what leads to heart attack. But I couldn't convince the reviewers. They kept killing it by saying, look, you need blood vessels to feed the heart, and you're giving this to. So this story was heard also for tuberculosis, and then Gates Foundation is tapped in because I couldn't get NIH to fund it. One thing I learned in science in my career is that if you move science by 5 degrees, you consider great. 10 degrees, even greater. 15 degrees, you're a genius. If you move it by 180 degrees, which is what I did, you're considered an idiot. So anyway, I've always had trouble because of these kinds. So but very fortunate, a young faculty member, not so young, but relatively, at MGH, just received a grant from NIH to test this idea. So hopefully in a year or two we'll set that up. But it's worth testing. Yeah.
Okay.
Because right now there's no treatment to block the rupture of, all the treatments are after what happens. Then you give anticoagulants, all these things, but that's too late by then. Yeah.
Well,
yeah.
thank you. So this is like in robotics, uncanny valley. You don't want to be too close to, a robot shouldn't look like humans. So, and rejection of grants is, Bob Langer always talks about it, right? That conventional.
He's a dear friend of mine. Yes,
and they reject it all the time. So I don't want to ask science question because then there'll be many. For engineering audience, how did you become such an incredible biologist, right? It's a switch from that. And perhaps if you have time, if you allow, your mentoring style, because the last scrolling list is already itself amazing. So maybe you can address those human questions.
So the human question is just, I would say, curiosity.
Engineering to biology in those days was different. Today there is biomedical engineering.
I'm not going to deny it. It was hard. And in my high school, I went to my first class of biology. And there, the teacher cut open a frog. And I'm a Jain. I had never seen anything. So I immediately switched to Sanskrit. And even though I had missed the first 3 years of Sanskrit, 7th, 8th, 9th grade, now imagine learning 3 years of Sanskrit, because then I was in 9th grade, so I had to do that over the summer, and it was very hard. But anyway, so that tells you about my biology background. And
mentoring style. I mean, how did you mentor all these people to such success?
Oh, because I first of all learned it, and the best way to learn it is to mentor. There's no better way. And the nice thing about young people, they haven't developed, skepticism like old people do. Yeah. So they're open-minded, and So, no, but you have to also understand, I have students from dozen disciplines in my lab at any time. It's only, some engineers, MDs of different flavor, biologists of different flavor. So I have those. I mean, my lab is like United Nations because I've had students from 30 countries, and a lot of them are now leaders in academia, industry, and government. Yeah.
Yeah, please go ahead for your next question.
Hello, Dr. Jain. Amazing talk. As a chemical engineer that's also made the transition, you should talk about how important transport phenomena, diffusion, thermodynamics, kinetics is also applicable to the tumor microenvironment.
Yes, it is. Yeah.
So I have a couple of questions very quickly. First one is, when are we going to see the amazing innovations that you have and the results of the clinical trials being actually implemented in clinical practice, or are they already being implemented?
I mean, as I said, the FDA approval, that means
So they are using, yeah.
Oh,
so,
yeah. These are, in 2014, the schwannoma treatment was approved by UK in the FDA. So, yeah.
Okay, and so beyond, the blood flow that you talk about in the tumor microenvironment, do you also see a role of spatial biology, understanding the location of cells with respect to each other, and AI coming to really expose that hidden circuitry of cancer playing a role here?
Mm-hmm. So again, it was a 20-minute talk, so I don't get into all the details. So what we do, you saw the spatial biology in real time is what that microscopy does. And on top of that, we have done spatial sequencing. And these are the new tools which have been developed. And we had a paper last year, Nature Genetics, on that. So yeah, we're using extensively genomics, genetics, and various bioinformatics approaches to do that. And AI is just beginning to come in. Yeah.
Yeah, thank you.
Yeah, Dr. Jain, it was a wonderful, wonderful presentation. I wish my son was here. This is very impressive. I had a question about the NIH funding because, someone like you with so much history of research, you had to fight for funding for 4 or 5 years because of a different way of thinking and the whole COVID, post-COVID stuff. Do you think sometimes we need to have 3, 4 types of research funding as opposed to you having to go to Bill Gates Foundation and other things? Because I always wonder, is science getting too centralized in the grant process?
Well, it is hard to get money. I am not going to try that. You think it's easy now after having all these publications? No, it's not. My grants still get rejected. And a lot of regions have stupid regions, but you just have to put up with it. It's part of the show, And it was very hurtful in the beginning when I was like 25-year-old kid writing my grants, and so I was expecting them to say encouraging words. They just killed it. But then with time you develop thick skin, and and actually you anticipate it's going to be rejected, and then you say, okay, I'm going to go back. So you have to start planning early enough, and that's what you have to do. And that's where the private foundations come in. And I've had money from the Gates Foundation. I get some money from small, but I have not had a billionaire sponsor yet, who would just simply write off my lab. And if somebody here is willing to do that, I'm ready. I'll approach Harvard, and they will put their name on that. It'll be Professor XYZ Laboratory for Tumor Microenvironment, or so and so. No, the Harvard is very happy to receive money for things like this. Yeah. So anybody wants to do the,
Unfortunately, we have a hard stop at 3 because
Okay.
there is a session afterwards. There is a hard stop at 3 because there is a session supposed to start. So Dr. Jain is going to be available after this session. There is another session coming on health.
Yeah, so I'll stay for that session, but I'll be around outside or in the dinner area or wherever we are, and more than happy to answer. And if you want, come to that 4-day course. Seriously, it's a virtual course. You don't have to go anywhere. Plus recorded and available for 1 month. So you can access that. But it's a really good investment if you want to understand this. The leaders who start.
Well, I guess there's some setup time between the two sessions,
yeah.
so let's keep it up. And this is a token of appreciation for coming all the way from Boston and enlightening us.
Thank you very much. Thanks again for the warm welcome and invitation.